Hello, everybody, and welcome to Beyond the Model, episode 7, which is actually the final episode in this series. It's been great. So, for anybody that doesn't know, Beyond the Model is a community run by Strativ Group. I'm your host today, Kieran Haynes. Beyond the Model, we talk about everything relevant to machine learning research, anything cutting edge, the latest innovations, the latest news, and the most important things. Today, I am joined by Jason Rolfe, who is the co-founder and CTO of Variational AI. Jason, it's great to have you on board, and thank you for coming. Today, we're going to talk about a variety of different topics, but as has been a theme with a lot of the stuff I've done, we're going to be talking about AI drug discovery. Jason's got some very, very interesting things to talk about. I can't wait to hear them. But yeah, Jason, welcome. How are you?
I'm excellent, Kieran. Thanks for having me.
Yeah, awesome. So, as always, Jason, let's have just a bit of a start. Get a bit of an introduction to yourself, who you are, what you're doing now, and we'll go from there.
Perfect. So as you said, Kieran, I am co-founder and CTO of Variational AI. I've been working in machine learning research for about 17 years now, focused primarily on generative models. During my PhD, I was looking at biologically plausible machine learning, trying to figure out what sort of algorithms the brain actually uses. The conclusion that I came to is that it was a form of a generative model. That was very interesting work, pulled me in some unconventional directions. And, that was my entree into the machine learning world. I did my postdoc with Jan LeCun, studying more conventional machine learning, and after that moved over to D-Wave Systems, where I developed machine learning algorithms that were compatible with their adiabatic quantum annealer. After building a fundamental algorithm that could make use of their hardware in a generative machine learning framework, I then turned to applications of that to try to tie the hardware through the machine learning algorithm to something that then someone would actually care about. Worked on a variety of applications, and in that process identified small molecule drug discovery as a really promising direction in which to apply generative machine learning. At some point, D-Wave decided that it no longer wanted to have a machine learning group. A number of people who were working there, where we already had a great team, a lot of very skilled and experienced individuals. We had identified a problem that was really interesting and decided to form a startup in order to pursue that. And that's what I've been doing with Variational for the past 5 or 6 years.
Yeah, awesome. Okay, first thing. I spend a lot of time speaking to people who do many different forms of machine learning research applied into the life sciences or one way, shape, or form. Just out of curiosity, when did it first hit you that this might be big? When did that first happen? Do you think you first realized some of the things that you could do?
I was looking into this again about 6 or 7 years ago. Looking in a variety of days, we were looking at model predictive control for industrial process automation. I spent extended period of time trying to train generative models on gene expression data. That was the first time I think I was working with a biological data set, which I'm sure we'll talk about more today. Gene expression data in particular is very challenging to work with. It's extremely noisy, and those generative models were disappointing in the sort of structure that they learned. I also was working on a project at D-Wave trying to separate environmental versus genetic causes of diseases, again, based upon sort of biological medical data sets. So I was thinking in that direction at that time, also looking into small molecule drug discovery. It's an area of exceptional import.
Yep.
Developing a new pharmaceutical fully capitalized is generally estimated to take about 10 years and $2 billion. A large part of that cost is because the vast majority of drug discovery programs fail. Most of them fail in early stage discovery. Even the ones that make it into phase 1 clinical trials, something like half of them fail. Then another something like half fail in phase 2, and then another third or half fail in phase 3. It gets exponentially more expensive as you move through that process.
Yes.
So the cost to actually develop a drug that is approved by the FDA and can be distributed to patients ends up being extraordinarily expensive. The time course between identifying the biological target that you're trying to modulate with the small molecule to actually having a drug that can be delivered to a patient, again, it's like a decade.
Yeah.
So there's a huge amount of unrealized value to humanity in being able to develop new drugs for the conditions that continue to plague us. Um, there's also the opportunity to create a lot of commercial value as well.
Of course.
So it seemed like a really nice direction to pursue. I
Yeah.
think it's a particularly interesting problem from the machine learning perspective because the there isn't the opportunity to solve this problem with big data. To collect one additional data point on a truly novel molecule, a molecule that is structurally distinct from those that have previously been synthesized and tested. It's a process that costs a few $1,000s minimum and takes 3 or 4 months minimum. As the molecule becomes harder and harder to synthesize, that cost in terms of dollars and time can go up exponentially to the point where there are total synthesis projects that consume an entire person's PhD.
Wow.
So this isn't a situation where you're going to be able to dig yourself out of it by just generating a million times more data than we currently
Yeah.
have. Even generating 10 times more data than we currently have is infeasible financially, unless there are techniques that can generate large amounts of data. They do so in extremely narrow corners of chemical space. So you get a lot of data points numerically, but they're not diverse. And so their overall impact is much more limited than you would expect from the sort of scaling laws that we see in natural
Right.
language or computer vision. Um, so it's not a problem that you can get yourself out of by just getting more data and throwing bigger models at it. From a research perspective, that makes it attractive because you don't need to be Meta or Google or Amazon to afford the compute to do
Right.
actual interesting research on this. You don't need to throw $10 million training each model. That big a model is not really useful on the scale of data that exists or will exist anytime in the near future. So it was also a really exciting prospect in that regard.
You know, I've actually heard this from a lot of people, and a lot of people, I think, probably similar to yourself and certainly some of the team at Variational and a lot of other sort of AI R&D labs looking at drug discovery, is that it really is okay. It's an important problem, an incredibly important problem for many obvious reasons, but it's such a difficult problem. It's such an interesting, because nature is so complicated. And,
It's it's unconventional from the sort of feature-based perspective as well. So when you
right.
look at computer vision, the standard understanding, which is true both on the machine learning side, but even on the biological neural side, if you're recording from neurons in the brain,
Right.
you can look at what sort of filters, what sort of features are learned at each level of the hierarchy. And at the bottommost level, you make edge detectors. And then you connect those edges into lines and curves. And then you build those into simple shapes and textures. And then you build those into objects. And there's been a lot of interesting work, both on the machine learning side and on the brain side, showing that
Yes.
actually sort of similar things happen in both cases. But you have this intuitive notion of what the basic constituent features of an image are for the purpose of human semantic recognition.
Yes.
Doesn't really hold in the case of small molecules. There are these things called activity cliffs, where you can make the smallest imaginable change to a molecule. You can add an extra methyl group, which is a carbon surrounded by hydrogens. People generally sort of ignore the hydrogen, so this is like adding a single atom onto the molecule. And that can change potency in some situations by tenfold or 100-fold. If the small molecule needs to fit into the protein extremely tightly. It's
Yes.
sort of like a lock and a key, except the lock is extremely flexible and the key is flexible, so they deform with respect to each other. And they're surrounded by water molecules, which are physically unconstrained but form extremely strong interactions. So you need to take all of those into account. And there are quantum mechanical interactions that affect the nature of the binding. But you need this small molecule to fit very tightly and with just the right sort of interactions with respect to the target protein that you're trying to bind to and either inhibit or activate. If you make a very small change in the molecule, potentially you shift that out of alignment. So the two would be physically clashing, overlapping. Obviously, two atoms can't be in the same location. All of a sudden, the orientation, which gave you such a great fit, such a tight coupling, now they're clashing with each other, they're knocking into each other, it doesn't fit like that, and you go from an extremely potent, effective drug to something that doesn't work at all. It's not consistently possible to identify something like edges within a small molecule to say, okay, here are the basic building blocks, and then we'll combine those together into some higher order structure and combine those together. There are functional groups which make sense in that sense from a chemistry perspective, but in terms of the pharmacology of the small molecule protein interaction, again, these tiny changes, the sort of things that would be a data augmentation that you want to be robust to in the case of visual data, these are the things that you need to be sensitive to in the small molecule drug case. So it's an unconventional, interesting problem, and a lot of the techniques that work so well in computer vision, like convolutional neural networks and data augmentation in particular, are not obviously applicable in this domain, which means there's the opportunity to develop something that's truly novel in order to solve these difficult but important problems.
I think you mentioned something really interesting, and it's how such a small on the surface to maybe an untrained biologist or somebody not familiar with the biology or chemistry itself, but such a small, tiny change can impact something so dramatically. And that's why, like, when this all changed, when AlphaFold was released, and subsequent iterations, and you sort of had this, like investors thinking, well, we can just design these drugs like super easy now, we're gonna fix this problem, and it just did not work, right? Because of what you're saying, and I think this sort of segues quite nicely into, what you and the team have been up to, and the sort of work that you do, and you spent a long time really researching into, and I think. Give us a bit of an overview then, Jason, of Variational. So, like the story, the narrative, how it happened, and I guess where you are now, we can dive into that a little bit.
Okay, so again, in terms of history, the founding team was all working at D-Wave Systems. D-Wave decided that it no longer wanted a machine learning department. There were a number of very highly skilled and experienced machine learning researchers there. We'd all been working together for years. We had identified an important problem, so it was really a perfect opportunity to come together and form a startup to address this problem. We had the team, we had the market that we wanted to go after. We had some, broad strokes ideas as to what the product would be, and we just took that and ran with it. Our initial stumbling steps, we were all initially machine learning researchers. Since we have hired cheminformaticians and computational chemists and medicinal chemists in order to ensure that there are adults in the room from the chemistry and pharmacology side to make sure we don't do anything too silly. We were very overly ambitious initially in terms of our expected timeframes for being able to predict and optimize over small molecule structures. We learned some lessons the hard way in terms of how difficult the data is. The size is not as big as what people are used to nowadays for like large language models. It's more like the size of ImageNet. We're training on the order of millions of experimental potency data points. But the thing that makes it, amongst the things that makes it challenging, the data is very sparse. For some targets, you have tens of thousands of experimental data points. For some, you have, down to hundreds or tens.
Right.
Obviously, we are much less confident about the output of the model if we only have 100 data points versus
Right.
tens of thousands of data points. But the data is also very noisy, both due to the nature of the experimental assays that are used to collect the data, but also because different assays are used by different labs. And all of this ends
Right.
up being aggregated together into one large data set because you want to use all of it. So, of course, you try to make use of whatever metadata is available to make the system aware of the fact that there are different experimental approaches being used to assess the same underlying physical phenomenon that you care about, but nevertheless, they're all subject to their own biases and idiosyncrasies. Even the same assay run by two different labs will yield somewhat different results. Um, so designing an algorithm that's robust to that is a challenge. Um,
Yes.
we need to deal with very imbalanced data. People are much more interested in active molecules than they are in inactive molecules for obvious reasons. It's the active ones that you're trying to develop into a drug. They also are very biased in their collection in that after they find something that shows some activity, they will make very small perturbations to that initial molecule in order to try to tune it into something that could become a drug candidate. But that means you get this series of tens or hundreds of structurally very similar compounds on which you have pretty good data, but you only have that data in a few corners of chemical
I
space.
see.
So it's not as valuable as if they were IID samples from the entirety of chemical space. Chemical
Right.
space is huge. People estimate it as between 10 to the 23 and 10 to the 60 synthesizable drug-like compounds. This
Oh,
is more
wow.
than the number of water molecules on Earth. You can't do a exhaustive search over that. Um, but the space that people regularly synthesize and test is a small corner of that. In particular, a lot of the early stage data either comes from high throughput screens in which people just have 100,000 pre-synthesized compounds, pre-plated, ready for like roboticized experiments, and they'll test the same compounds against every target. You can just buy these things from commercial vendors
Oh,
in order
wow.
to do your experiments. But that means that you're looking where the street light, you're looking for your keys in the dark underneath the street light. You're looking at the compounds that have already been synthesized, and everyone's sort of starting from the same point rather than trying to push out into the more novel regions of chemical space.
Yes.
The other more recent approach is based upon make-on-demand libraries, in which you have a set of 100,000, hundreds of thousands of starting fragments and a few simple, reliable reactions to put these starting fragments together. And you want to do, like two or maybe three reactions to get from your starting fragments to the final molecule. And this opens up a numerically larger space of, I think they're up to like maybe hundreds of billions now, which again, it sounds really big, but compared to 10 to the 60, it's still
Yeah.
0%.
Yeah.
And because it's the same very limited set of reactions on a fixed set of starting fragments, it ends up focusing you in this very narrow corner of chemical space. Which, again, like if you can find a drug candidate that's close to there, it's a very efficient way of proceeding. From a machine learning perspective, it's not the problem that you're used to trying to solve because the data is not IID from the full space of synthesizable drug-like molecules. And you really do want to be able to extrapolate as far as possible in order to find the novel chemical matter, which is going to be able to address conditions that have not previously been able to be addressed. Also, you want to be able to patent your drug because it's not commercially viable to develop a drug if you can't patent it and then profit
Right.
from it. So drugs won't be made if they can't be patented. And the patents are usually tied to composition of matter, and they need to be sufficiently structurally novel in order to get the patent, which then
Interesting.
unlocks the ability to actually develop the drug through financing and eventually getting some sort of monetary return on your investment.
It's I've so before machine learning was used in drug discovery, like I've heard it compared to I've even heard biologists say this, that it's almost like it was like an art for them, right? Almost like intuition, right?
It still is.
Yeah.
So there are experiments. The people who do this process of tweaking the small molecules are medicinal chemists. And
Right.
there have been experiments done in which you give 5 or 10 or 20 medicinal chemists the same starting information and say, like, which molecule should you look at next? And the medicinal chemists will all give a different answer. And most of them will be very confident in their answer. They'll say, like, oh, I had an experience with a similar compound before, and, like, we added an extra fluorine over here, and it was fantastic, so you should absolutely do that. And the next one will be like, fluorine? You shouldn't put a fluorine there. It's very much a dark art.
A dark art, exactly. It is a dark art, and this is the thing as well, because you said, okay, 10 to the, what was it, 10 to the 60? Is that
That's the upper estimate?
Right. I mean, that is an insanely large number. Currently, of course, we can't explore. Maybe in the future, maybe in the near future, who knows? But like you said, right, there's got to be a different approach. And I suppose with the team Variational, you have Enki, which is the model that you guys have designed and built. Firstly, just tell me about Enki. Tell me about why you why you built what you built, and what sort of what sort of real impact it can have.
Okay, so Enki is a generative foundation model over molecular structures and molecular properties. A generative model is a natural fit for this problem. Like 10 to the 60 is obviously a really big number. It's not larger than the number of natural images that you could put together by choosing individual pixels in your image. It's not bigger than the number of paragraphs that can be written in English.
Oh, wow.
These are exactly the spaces. I wouldn't want to have to estimate those numbers off the top of my head. It would require
Big
5
numbers.
minutes and a napkin, but those are also astronomically large numbers. It's a combinatorial explosion because you get to independently choose each pixel or word. It's obviously not independent because there are dependencies which make it a well-formed image or a grammatically and semantically meaningful sentence. But nevertheless, there is a huge space there. Generative models, foundation models, have proven very successful in
very good
producing novel instances of images and text. So it would be natural to say, we should apply this to molecules.
Right.
There's obviously a number of challenges there. The first challenge that you run into is synthesizability. When you're training on natural images or on natural language, you don't train on snow from your television. You don't train on random pixel combinations. You don't train on random strings of words. We have lots of pictures. We have lots of human-generated text to train on. The first complement to that on the small molecule drug discovery side are compounds that have been synthesized or are synthesizable.
Yes.
There are reasonably large data sets of that, but then you start restricting to drug-like. When people talk about drug-like, they're worried about things like oral bioavailability. When you stick a pill in your mouth, the molecule needs to be absorbed through the digestive tract. It needs to be transported to the site of action, not immediately filtered out by the liver, for instance, or the kidneys. It needs to be able to get it into the cells where the proteins that you're trying to modulate are located. It needs to be removed from those cells and then eventually metabolized and excreted so it doesn't build up to toxic concentrations. It can't interact with anything other than the target of interest. It can't be toxic. These are the sorts of things that go into drug likeness. And now the first problem you're going to run into is that even if you have a sufficiently large and diverse set of synthesizable molecules to train on, which even that's in question, but even if you had that, most of those compounds you would not want to use as a drug. They don't satisfy these drug likeness criteria. In order to use generative machine learning to produce a model that can produce synthesizable drug-like molecules, you should be training on synthesizable drug-like molecules. So that's definitely a challenge. And then beyond that, now we want to optimize within this space in order to find compounds that are potent against a particular drug target, a particular protein that a biologist has identified as being problematic, that you either want to, usually you want to inhibit, make it less active. Sometimes you want to activate it and make it more active.
Yeah.
But you want to have that impact on that one particular protein. You don't want to impact anything else. And
Yes.
frequently there are families of tens or hundreds of structurally related proteins, evolutionarily related proteins, and to interact with exactly one of them is a significant challenge. Um, so you need to train this generative foundation model not only over the molecular structure, capturing synthesizability, drug likeness, toxicity, absorption, distribution, metabolism, excretion, but also all of these other pharmacological properties of interest, its interaction with all of the possible drug targets, and then you need to be able to condition on that when you're doing your sampling procedure.
So it's a real dark art indeed, even from a machine learning perspective. And so, okay, and what are the things you look? That's a big challenge. You said at the start as well, the average cost to develop a drug, $2 billion. The average amount of time, 10 years, and in some cases a lot longer and a lot more costly as well. There's a reason this is hard, and this costs so much, right? And that this takes such a long amount of time. But you mentioned, obviously, foundation model, right? And in the broadest sense, right, even from my perspective, a foundation model being trained over many different data types and, not just completely specific examples of data, something to perform different tasks. What I'd be interested in understanding is firstly, what does that actually mean for Enki? To be a foundation model. And secondly, what's hard about training a foundation model in the context of small molecule drug discovery?
Okay. So there are various things that you could call a foundation model. If
Right.
you were just training over chemically stable molecules, you can get enormous sets of chemically stable molecules.
Right.
And there are data sets where people have enumerated all of the possible molecules made up of particular atoms up to 18 non-hydrogen atoms. Like this is a standard data set that people use in some aspects of computational chemistry. That would be a foundation model. It's not a foundation model that's particularly useful for drug discovery because it's not constrained to synthesizable
The challenges of drug discovery.
drug-like, let alone the pharmacological properties
Yeah.
that you care about. When we say foundation model, amongst the key things is that we are jointly training on 1,000 different targets.
Right.
So we can leverage transfer learning between those targets. Again, there are these families of related drug targets. They're structurally similar. Often similar molecules strongly bind to and either inhibit or activate them. You need to learn about this both in order to engineer in selectivity, so you inhibit exactly one target, but also just knowing about one of these targets informs you about another target. If you know what Great Danes and you know what Huskies look like, that helps you identify dachshunds. So we can utilize transfer learning both within target classes, but even across target classes. The nature of the ligand-protein interactions, the small molecule drug-target interactions, it's the same basic processes across target classes. You're looking at hydrogen bonds, you're looking at hydrophobic interactions, you're looking at pi stacking. The same basic interactions are operative in all cases. So there's the opportunity to make use of transfer learning there. And the foundation model, that is taking in both the structural aspects, the chemical stability, synthesizability, drug likeness, as well as the pharmacological properties, the potency against various targets, both within a single target class and across target classes. Pulling all of this into a single model enables you to make best use of the limited experimental data you have on any given target. So the analogy that I would draw would be to ImageNet. ImageNet is still one of the standard, it's no longer really a large data set. It's about 1 million images, 1,000 different classes, about 1,000 images within each class. If you only had 1,000 images from a single class and that was your full data set, you would not have a very strong model. But because you know about 1,000 different classes, each with a moderate amount of data, you learn just the structure of natural images, you learn the structure of objects, and you can do well on all of those classes, even though the data for any one class on its own wouldn't be sufficient. So a foundation model, a joint model over all of those targets is required there. And it's also what we're leveraging in our case to address the limited amount of data available on any given biological target for small molecule drug discovery.
That's awesome. And a question from me. This might be a silly question, so bear with me if it is. So one of the things that you mentioned was like the example in ImageNet, a husky and a Great Dane, right? One of the things that I was thinking when you were talking was going back to if there's the tiniest change in molecular structure, like you mentioned even an atom, right? It can wildly affect what happens. And the thing about designing drugs is you have to be so specific because you don't want any unintended properties. All of the things that you mentioned, the drug likeness and the pharmacological aspects. The question is it harder to train a foundation model when the classes, the class types here have to be so precise? Like, does that present different challenges as opposed to dog, cat, ginger cat, black cat, or does that not really come into play from a machine learning perspective?
It almost certainly makes the problem harder. And
Right.
Again, it requires you to use different architectures. So for instance, a convolutional neural network where you're basically dragging a spatially localized filter across the entire image is designed to detect an edge of fixed width and fixed length and fixed angle anywhere in the image. It's intuitive how it would do that. It's a great inductive bias. It enables you to learn a very powerful model with less data because you've matched your inductive bias to the data so well. I want to emphasize that in the small molecule case, it isn't the case that every single atom change induces an enormous change in the pharmacological properties.
But it
just
can.
that there do exist
Yes.
these very small structural changes that induce very large changes in the pharmacological properties, and it is rare in an image that if you change a single pixel, it completely changes the meaning. It requires that you find different sorts of features and that you have a different architecture to complement the relationship between the lowest level features of your input and the higher level properties that you want to predict and optimize. So it's definitely a challenge. We have found it not to be an insurmountable challenge.
Right.
I'm sure that there is a lot more work for us to do, and there are a variety of exciting algorithmic directions that we are presently pursuing in order to better align the inductive biases of our model architecture with the problem that we're trying to solve. But that's part of what makes this problem so exciting, that there is the opportunity to develop these new architectures to address this unconventional problem.
This is a really good point and a really interesting answer because, part of the work I do is help people go from a lot of the time a complete CS or ML background, right, from their PhD into working on biological problems. And you've seen a shift in journals and conferences over the last 5 or 10 years. And, NeurIPS, iClear, ICML now, and for quite a few years, there are dedicated bio workshops, there are more papers relevant to these things. And it's because of these are such difficult problems. It kind of enforces, in some cases, innovations in machine learning itself. You have to change how you're doing stuff and you land on new things, which can push the entire field of AI forwards. But it's such an interesting problem and it's so hard, It's becoming more and more exciting to work on, and as generative machine learning advances, as many different areas and modalities of machine learning become more sophisticated, you can tackle these problems in different ways. And I think that's part of what draws me onto the exact next question, right? And it's kind of like a mixed question that puts together two, I think, really important things, which is the impact of Enki for sure, but AI. What's realistic in AI drug discovery generally, and I suppose that to go to the former, what impact can Enki have for companies and for for biopharma companies, for pharma companies? How much can it help them?
I think it can be extremely impactful. It can reduce the time required to develop a drug candidate from years, potentially down to months.
Wow.
Save enormous amounts of money, increase the probability of finding that drug candidate. We've had a number of very successful commercial deals already. Our largest customer was Merck Research Labs. We did a project for them that they liked the results so much that Merck Global Health Innovation was one of the investors in our most recent round of funding.
Yeah, awesome. And pushing that on a little bit further, that's really awesome. And again, one thing that is always so interesting to me, even global giants like Merck, AstraZeneca, a lot of these big pharma companies, something that's pushing them on a lot so far is companies like Variational, right? And the myriad of AI drug discovery startups really pushing the boundaries of how creative you can be with machine learning. 10 years to months is a wild difference.
I want to emphasize it's not months for the entire drug discovery
Of
process.
course.
You still need to go through clinical
Of
trials.
course.
Clinical trials are also a very interesting potential application of machine learning. For instance,
Yes.
trying to match individual patients with the trial in order to maximize the observed impact, the statistical power of the analysis. To identify the particular sort of assays and readouts that are used in the clinical trials. That's another great application. It's not one that we're working on. There are other companies who are also pushing that, and that will also have a big impact. We are focused on the preclinical drug discovery,
Yes.
but that is also a years-long process, and we want to reduce the time and cost of that by many multiples.
You're taking it from a dark art into, a real bit of science, and that's kind of like what this is doing. And look, I ask everybody this question, and I think about it all the time, but you've kind of just hinted on it there, the impact that AI can have in drug discovery. And I suppose just from your own point of view, what you think, there are many impacts it can have. There are many ways it can be impacted. AI can change this in a lot of different ways. But what do let's just over the next 5-10 years, whatever, what do you think is actually realistic? What sort of things do you think we'll see in AI drug discovery?
Within 5 or 10 years, I do expect that the time required to move from drug target to drug candidate, knowing what protein you want to inhibit, to having a small molecule that, at least from the preclinical evaluation, seems to satisfy all that criteria. I do expect the time and cost of that to go down by 50%, 70%, 90%. The machine learning will get better and better, and it will rapidly accelerate that process. It will enable people to reach further out into novel chemical space, find
Yes.
molecules for targets that were previously undruggable. It will take into account more of the optimization criteria earlier in the drug discovery process. So selectivity in particular is a huge problem in developing a drug, because if the molecule isn't sufficiently selective, if it's hitting unexpected targets, that ends up manifesting as toxicity after you get into
Yes.
clinical trials.
Which is bad.
It's difficult. The problem is that there are so many molecules, so many proteins and other systems in the body that a given small molecule can interact with. You can't test for all of them in the early stage preclinical testing. And you observe it, if you're lucky, when you start doing animal testing. And if you're unlucky, you observe it when you start putting it into humans. A foundation model can model and predict and optimize against a much larger set of those possible targets and off targets in the case of any particular drug, and those interactions. And my hope and expectation is that will increase the probability of clinical success, because you are just starting from a better point. You have computationally taken care of things in the initial development of what's called a lead, the compound that you're going to do the fine tuning on. You've just found a better region of chemical space that is more suited to the particular pharmacological indication that you're going after, so that these surprise problems are less likely to come up when you move into clinical trials. It means that we're going to have more drugs, we're going to have cheaper drugs, we're going to have drugs against diseases that we did not have drugs for before, and it will be great for human health.
If you think about when drugs were being designed in the 50s, 60s, 70s, 80s, 90s, right? Even up until super early 2000s, but I think more, 70, 80 years ago. And you were to ask those people, you were to give what you're doing, you were to give Enki to these people, explain it, and the leaps and bounds of technological innovation that have happened between now and then, it would have just been, unthinkably crazy. You're generating these things via machine learning. And it always makes me think about what's ahead, like in 20 years, in 30 years, or in the next, I don't know, huge technological leaps in computing, whether it's quantum computing, whether it's a different type of machine learning. I think it will be, I personally think this is the most important area of machine learning, full stop, because it has the impact to really just rewire the course of human history and future as well, and how, and solve some of the biggest challenges that people face, like full stop. And it's so exciting that companies like Variational and people like yourself are doing what you're doing, and that more companies are popping up in this space, and that there are many different types of companies in across all the different areas of machine learning for drug discovery. So that's really cool to hear. And even in the next, I actually, just really quick, I saw it was a Canadian research company that put out a study in 2021, 2022. I can't remember who it is. I actually quoted this last episode as well. They reckoned that by 2030, all drugs that are created after 2030 will be in some way, shape or form designed with the help of or completely by AI or machine learning. And what an exciting future to look forward to. But I guess the last question for me, as is something we do on this series a lot, is actually from our previous guest, Nicole Haas. And he said what he was really interested to know is, well, why do you do what you do? What sort of keeps you going? So, Jason, why do you do what you do?
I want to make an impact on the world. I want to discover and develop something surprising and new and impactful and interesting. And this seems like the particular problem and domain in which I can have the highest chance of having that big impact, of moving the needle on something that's important.
What a fantastic answer. So, okay, given this is the end of the series, episode 7, we don't have a question for another guest, but that was awesome. I just want to say thank you so much for that. That was so interesting. I think everybody listening will really find that quite engaging on many different levels. We discussed a lot of really interesting things. Thank you very much, Jason, for coming on Beyond the Model. And yeah, it's been an absolute pleasure.
Thank you, Kieran. It was great to talk to you.